Welcome to this discussion over the muscle tissues. This represents the fourth and final lecture involved in exam number 2 in the course. Looking at the table of contents, today we're only going to be looking at 10.1, 10.2, and then we are going to jump, and even all of 10.2 will not be covered. And then we're going to jump down and look a little bit at 10.7, cardiac muscle, and then a little bit again at 10.8. We'll also continue on and look just briefly at the very beginning of chapter 11 in the first part of section 11.1 and in naming muscles and different muscle groups. There's also a few PowerPoint slides that I will refer to, and those are also available to you in your Blackboard shell, but they're also posted, and we'll be looking at those slides as well. So let's get started into muscle tissue. So we've been spending a lot of time looking at tissues, looking at histology. Since Lab 2, we spent a lot of time looking at 20 different tissues under the microscope. And we spent most of our time looking at epithelial tissues and connective tissues. You know the characteristics of those. You know the 7 different epithelial and the 9 different connective tissues that were presented. We also at that time did look at muscle tissue. Recall there are 3 types of muscle tissue: skeletal, cardiac, and smooth. And part of today's presentation will be really going back and reminding you of things you already know, with a couple additional facts added in about these 3 different types of muscle. So muscle, again, is one of the 4 primary tissue types. And let me just remind you that we learned about the characteristics of epithelial tissues. And you really should be able to just list these characteristics off. Epithelial tissues are composed of tightly packed cells with little to no extracellular matrix. Epithelial tissues are avascular. Epithelial tissues sit on a basement membrane with connective tissues below. Those are the main characteristics. Remember, too, the main characteristics of connective tissues. We have loosely arranged cells with lots of space between the cells, therefore lots of extracellular matrix. These tissues are surrounded by a ground substance, and in that ground substance there are protein fibers. Oftentimes we see collagen, elastin, and reticular, but when we're discussing blood in the plasma, we see a different collection of proteins. But regardless, loosely packed cells surrounded by ground substance and protein fibers. Those are the common characteristics found in all of the connective tissues. So now we get to muscle tissue, and you shouldn't be surprised. There are characteristics that are going to be common between skeletal muscle, cardiac muscle, and smooth muscle. And the first of these characteristics is that all muscle tissues are excitable. Excitable. That means that they are going to get excited. This will happen from the nervous system. The specifics of how that works will be a story that you will learn in detail in physiology. But just know that the nervous system is going to send a signal down a nerve to the muscle, and that is going to cause the muscle to become excited. Now, once that muscle becomes excited, that muscle is going to contract. That's what happens, right? Muscle cells are excitable. And then when they get excited, they contract. That's just a fancy way of saying that they're going to get shorter. Muscles are going to get shorter. They're going to change shape. Well, when they change shape, they also exhibit a third characteristic in that they are elastic. You know what a rubber band does? A rubber band stretches but then recoils back to its original shape. So even though muscles are going to change shape, they're going to recoil. They're going to return back to their original shape. And then finally, and this is probably the one that is the most challenging to appreciate is that muscle cells and muscle tissues are extensible. That is, when you are lengthening, or I should say, when you are shortening a muscle, when you are contracting a muscle, there are usually opposing muscles that are at the same time becoming longer. A good example of that is if you right now just flex your biceps brachii, flex your elbow. So contract your biceps brachii. And as you bring your fist closer to your shoulder, your biceps brachii muscle is shortening, but your triceps brachii muscle along the posterior side is actually elongating. And so that is that extensible idea that the opposing muscle group is going to be extending or stretching. So the four characteristics of muscle tissues, all of them are excitable, they are contractile, they are elastic, and they are extensible. Let's quickly review the histological image of these three tissues. So right now we're looking at skeletal muscle. You've seen this before. You recall that these are very long cells. We call those cells also fibers. We can see in the micrograph the strong striping pattern. We are also noticing that there are many nuclei along the length of that muscle cell, and that so these are considered multinucleated cells. And we see that they're long, skinny cells that are running way beyond even the view that we're seeing on the screen. In this particular micrograph, we are seeing essentially or approximately 1, 2, 3, 4, 5, 6, 7, 8, 9 cells running from left to right across the screen. So that's skeletal muscle. Go secondarily to cardiac muscle. Again, you've seen this micrograph before. We're looking at Y-shaped cells. Look at the branching. You can see the branching in the tissue. We don't see the multinucleated presentation. Cardiac is mostly uninucleated. And while this picture doesn't have intercalated discs quite as distinct as I'd like to see, there are these intercalated discs along the way. They're just not quite as strong as we'll see in some other images. And those intercalated discs are a telltale sign that you are also looking at cardiac muscle. So if you see the branching pattern and you see intercalated discs, you are definitely looking at cardiac muscle tissue. And then lastly, let's remind ourselves about smooth muscle. And in this particular micrograph, you don't see the strong striping patterns, you don't see the striations. That's a characteristic. The cells and the nuclei are these long football or cigar shaped looking cells, and that is another characteristic of smooth muscle tissue. So all three of these are excitable. All three of these tissues contract, they shorten. All three of them return back to their original shape, and all of them are extensible as well, in that they stretch and return in the opposing directions. Another thing we know about muscle is that we've discussed how muscles all require a lot of energy. So there's a lot of ATP. Well, any tissue that has a lot of ATP, you can expect is going to have a lot of mitochondria. So we're going to see a fair number, a greater number of mitochondria in muscle tissue compared to other tissue cells. And I've mentioned briefly that calcium is also a critical ion for muscles to contract, and we'll talk about that interaction a little bit more later on. I want you to know about the modality of muscle, that is the tissue voluntary or involuntary? And we've been working through this enough. You now recall that skeletal muscle is voluntary. It takes your conscious movement of your muscles for them to move. You have voluntary control over them. Whereas cardiac muscle and smooth muscle is involuntary. These are muscle types that work 24/7. You can go to sleep. You do not have to consciously or voluntarily control those muscles. So we refer to them as involuntary. You also know in our comparisons that skeletal muscle and cardiac muscle are both striated, right? We have the striations in skeletal and cardiac. So make sure that you are thinking about how these muscle tissues are similar and how they are different from each other. In this cartoon, I hope you look at this and can immediately recognize the Y branching, the branching of these cells, and you see the striations. And so you should quickly recognize that this illustration, this animation represents cardiac muscle. Do watch these animations to give you a greater and better understanding of these tissues. Here's a great little animation on smooth muscle. So I'm going to jump over to the accessory slides. We're at the end of 10.1 in Blosson. Don't forget to answer these questions. These again will help you compare and contrast the different types of muscle tissues. But let me jump over to our set of slides for a moment. So these slides are also posted in your Blackboard shell right underneath this video presentation. Just briefly, there are over 700 skeletal muscles in your body that have been named. Okay, all of these muscles make up your muscular system, and they are all over the body. You can consider muscles the organs of the muscular system. Of course, we're moving things. In addition to moving our skeleton, we're also, with the help of the cardiac muscle, moving blood through our body. And when the help of the smooth muscles, we're moving digestive products, for example, through the digestive system. Each muscle is composed of all four tissue types. So if I give you a chunk of skeletal muscle or any of the muscles, you should now be able to show me very clearly where there is, in addition to quote muscle tissue, but where I also see some epithelial tissue, where I see some connective tissue, and where you see some nervous tissue. A single muscle is composed of thousands and thousands of cells. Each cell is itself called a muscle fiber. And as we're talking in this particular slide, mostly about skeletal muscle, when you see a muscle, the cells in that muscle, the cells are as long as the muscle is. And so if you have a 12-inch muscle, the cells in that muscle are also 12 inches long. And recall that skeletal muscle is multinucleated, simply meaning that those myoblasts, that word should make sense to you, the cells that make the muscle initially, they all combine end to end into those long fibers. And for reasons that I'm not sure about, but the nucleus is maintained. And so that's why we see multinucleated skeletal cells, skeletal muscle cells. Now, I want to bring up this cartoon representation. Again, it's a great way of easily and quickly comparing and contrasting the three muscle types. On the top, we're looking at skeletal muscle. Again, we see the long, skinny cells. We're looking at glimpses of 3 cells in this particular cartoon. We're seeing lots of striping patterns. We're seeing multiple nuclei. If I were to discuss the characteristics of skeletal muscle, I would say that skeletal muscle is strong, it's quick, and it's voluntary, and it's discontinuous. We've talked about voluntary before, but discontinuous simply means that you could have your arm all night just sitting there, right, sleeping, or you could just have your arm hanging down by your side all day, and you don't have to be continuously flexing your arm or contracting your muscles. So skeletal muscle is discontinuous. You're only going to contract the muscles when you need to. Whereas in the middle, we have cardiac muscle. And cardiac muscle, again, we see the strong striping patterns. We can see the branch patterns. And we also see in this rendition, we see these lines, those zigzag sort of etch-a-sketch looking drawn lines are the intercalated discs. This muscle, cardiac muscle, is also strong. Your heart is a very strong muscle. It is also very quick. It contracts 60, 70, 80 times a minute, but it is involuntary and it is continuous. We don't have an option here, folks. Our heart must continuously contract for life to be sustained. And then lastly, on the bottom, we have smooth muscle. And you can see that the cells are much, much smaller. We don't see any striations. These cells are long and little fusiform shaped cells, these little cigar shaped cells, a single nucleus. And if I were to describe smooth muscle, I would say that it is relatively weak and relatively slow. This is involuntary muscle, and we find it as a great example lining the walls of the intestines and stomach as we process our digestive. So we'll come back and look at these other slides in a moment. For now, though, let's just jump back into our blossom. So the majority of our conversation today is going to be about skeletal muscle. So everything I'm about to say is referring only to skeletal muscle. Again, we'll be comparing and contrasting skeletal muscle with cardiac and smooth as we continue through this. We are not going through this entire section. We will be skipping a little bit at the end, and I'll make that very clear. You know some of the characteristics of skeletal muscle. Skeletal muscle is largely attached to your skeleton. It is allowing us to move our skeleton, hence its name. It is also skeletal muscle that is contributing to the movement of joints as our bones are changing orientation. Skeletal muscles are also generating heat. When you are moving your muscles, you're generating heat. When you are cold, your body might instinctively or through reflexes begin to shiver to create more heat. Now, skeletal muscle, a skeletal muscle is an organ. It is made up of many, many, many muscle fibers or muscle cells. But of course, all of those muscle cells must have a direct connection to the blood supply and to the nervous signals that are generating the contraction and the excitability. And there's also, we're going to see, a fair amount of connective tissue that helps to bind a muscle together into the organ that we see. So let's now turn our attention to the three different layers of connective tissue. These can be referred to as the mesia or the mycia, you'll hear it pronounced both ways, that are going to provide the compartmentalization for the structure of the muscle cell. And the first of these is called the epimysium. Now, in this chapter, we see a lot of myo terms, myo this, mesia that. And so we see mesium or mycelium, and so that is a connective tissue layer. Epi, we know, means on or upon. This particular layer is made up of a dense, irregular connective tissue, and epimysium is going to be on the entire muscle. So let's take a look at this, and let's get our bearings on this image. So this is back here a skeletal muscle, and in the purple fuchsia kind of color around the entire muscle, it's labeled here, that is the epimysium. So that's the layer of connective tissue found all the way around your entire muscle. Now, when we cut into a muscle, as we have here, we notice that the muscle is composed of these sections, these very bundled sections, and each of those sections is called a fascicle. So the muscle itself is full of bundles. You saw in your vocab that fasci means band or bundles. And those fascicles are surrounded by a second type or a second layer of connective tissue referred to as the perimysium. You know, peri means around or perimeter. So this is a type of connective tissue that goes around the muscle and is one of the connective tissue layers. Let's take a look at that image. So here we see this is the entire muscle. Pulled out of that, we see this entire big structure. This is a fascicle, it's labeled. And the green layer going around it, the outer layer here shown, is the perimysium. So the perimysium is the connective tissue layer. If we look back, we can appreciate now that what we're seeing in the picture as I'm pointing around it, that lighter color is the perimysium. Notice too that there are these connective tissue layers are providing a space for the blood vessels and the nerves to travel down into the depths of that muscle. And that's important for the supply of oxygen and nutrients and hormones, getting rid of waste products, and of course also the muscle cells need to be in direct connection with the nervous system, and so we need nerve fibers that also go deep into the muscle tissue. Now, every muscle fiber itself is also covered by a layer of connective tissue referred to as the endomysium, and of course that makes sense to us. Endo means within, so deep within the muscle tissue there is the endomysium. So let's look again at what we're seeing. This back here is the fascicle, and we see one cell pulled out here. This is the muscle fiber. We'll look at the cell here in a second. And so this is this fuchsia teal color. That is the endomysium that is surrounding each and every individual cell. Again, notice that these connective tissue layers are providing a space for blood vessels to be very intimately and very deeply connected into the muscle tissue. And then here's a muscle fiber, and we'll take a look at some of those special characteristics in a moment. So coming down here, we see if this is the muscle cell itself, and the muscle cell itself is full of a specialized organelle called a myofibril. Now, myofibrils, that's a new term for us. We learned about a lot of different cellular organelles back in chapter 3, the Golgi, the endoplasmic reticulum, the nucleus, the centrioles, the lysosomes, etc. Myofibrils were not introduced back then because myofibrils are not found in all cells, but are a specialized organelle found in muscle cells. And they run the entire length of the muscle cell. As you can see, the muscle cell is largely just filled with these elongated myofibrils. And as this myofibril is pulled out here from the cell, you start to see this light and dark banding, and you're beginning to perhaps get a glimpse of why it is that skeletal muscle is striated. There is a light and dark banding pattern, and again, we're beginning to see evidence of that in this myofibril. Also labeled here for the first time is the outer layer of a muscle cell, and muscle cells are just different cells. They're so unique. So there is a separate vocabulary to describe the pieces and parts of a muscle cell. And the outer layer of a muscle cell, you might want to call it just the cell membrane, but it's specialized and it's called the sarcolemma. Sarco, in addition to seeing a lot of myo terms in this unit, we're going to see a lot of sarco terms. Sarco means flesh, muscle, or flesh. When you're eating a steak, you're eating the flesh, right? The muscle of an organism. And so the sarco means flesh or muscle as well. Lemma is a term that means rind or peel, the outer layer, like a watermelon rind around the outside of a watermelon. And so sarcolemma is the outer layer of the muscle cell. It's highlighted here in sort of this yellow tinge. So I'm going to jump over to some other pictures here in a moment, but to reconnect, we have three layers of connective tissue from superficial to deep. There's the epimysium, the perimysium, and the endomysium. And let's jump over to a couple other slides in the PowerPoints to show these images once again. So looking at this particular image, we can see the three layers of connective tissue are shown. We're even putting on a little bit more detail in this picture. This is why I wanted to give you this for orientation. So we're looking at a long bone here. I'm seeing a very large head, and I see a big bump here. This is the greater trochanter. This is the lesser trochanter. So this is a femur. And so this is a muscle attached to the femur. And if we were to just think about our tissues for just a moment, there are quite a few tissues represented here, aren't there? If I were to take a little bit of this cartilage from the head of the femur, I know that that is hyaline cartilage. I know what I would expect. I would see chondrocytes in lacuna. Then if I took some compact bone, I know I would see osteons and canaliculi and lamellae and central canals. As I look at this, I see a tendon. A tendon, remember, is dense, regular connective tissue. And that tendon is connected from the bone to a muscle. So we see this is a muscle. This whole thing, muscle, is going to be surrounded by, we're zooming into this little box right here. And so this little outer layer around the entire muscle is the epimysium. Inside the muscle, I see bundles. I see fascicles. Each fascicle is surrounded by this lighter, sort of periwinkle looking colored tissue. That is the perimysium. And then we know that the individual fascicles, here's one fascicle pulled out, that they are full of cells. And so here is a muscle cell. The artist has even put on it two nuclei. So we know we're looking at a multinucleated muscle cell. And each of the muscle cells would be surrounded by a layer of endomysium. In addition, we see one additional layer here, fascia. Fascia is a connective tissue layer that connects muscles to other structures. So here the deep fascia is a continuation of the tendinous material. Let's just look at these again in a different view. Now, this bone, what bone is this? I don't see quite as large a rounded head, but it definitely is a head structure. This is the humerus. And so this would be the long bone of the glenohumeral joint. And the two bumps here, the tubercles, there's a greater and the lesser tubercle. And here is the tendon coming down. Again, that deep fascia coming around the entire muscle. Then we see the entire muscle is surrounded by a layer of epimysium. Inside we see fascicles, it's all labeled. The fascicle is surrounded by perimysium. Again, we see spaces for blood vessels and nerves. The muscle fiber is here. It is surrounded by endomysium. The muscle cell here, we see again a couple nuclei. A reminder that this is at the cellular level, at the fiber level. And we see in this rendition that a myofibril has been pulled out, and we get a glimpse of those striations, those light and dark bands. And for the first time, the cell here in the very bottom, there's a yellow cell here. This is a satellite cell. We'll see this again in a moment, but a satellite cell is a stem cell in muscle tissue. So if you damage a muscle, there are stem cells waiting around for repair mechanisms. Think osteogenic cells are in bone, waiting for you to break a bone, and in muscle, there are satellite cells, as the name suggests, are around the outside of the cell, and they are there to help regenerate a damaged muscle cell. Well, in this picture, I mentioned the deep fascia. If there's a deep fascia, there must also be a superficial fascia. And you already know the term superficial fascia. It's not shown in this particular image, but you'll see it in future images. But you already know the superficial fascia by the name subcutaneous layer or hypodermis. So the superficial fascia is your hypodermis. You know that's mostly fat with some areolar connective tissue that separates muscle from skin. So superficial fascia, aka subcutaneous layer, makes sense, subcutaneous under the cutaneous membrane, or hypodermis under the dermis. All three of these are interchangeable terms. So let's look at that superficial fascia a little bit more closely. Here we're looking at a transverse section through the femoral region. And in your thigh, you are learning that there are a lot of muscles. In lab, you're seeing a lot of muscles. Your quadriceps group, your hamstring group, and a number of other adductor muscles that are traveling through the thigh. And so this slice to me looks like a ham steak, does it not? That you have all of this muscle with the ham bone in the middle. There's your femur. And around it, there's a layer of fat. That layer of fat is the superficial fascia, aka the hypodermis, aka the subcutaneous fat. And you can see that all those muscles in your thigh are separated by these peri and endomesial layer, or epimesial layers, right? So each muscle is able to slide over the other next two muscles, the adjacent muscles, because they are separated clearly by those epimesium and deep fascia layers. So if this is a muscle fiber, but how would you know that? If it wasn't labeled as muscle fiber, what other clues do you see in this picture that would help you determine that this is down at the cellular level? And the clues are as follows. The layer that's torn out and labeled here is the endomesium. The individual structure being pulled out of the cell is a myofibril. I see that because of the striping pattern. And I see the satellite cell here. All of those are clues, along with the multinucleation of this cell, that this is at the cellular level. What if instead I saw this image and it did not say fascicle? And you would know that it's a fascicle because it does label that there's a perimesium surrounding that entire structure. And here's the muscle cell. Again, we see multinucleated. We see evidence of the myofibrils in here. And we see the satellite cell once again. And then if this wasn't labeled as the muscle, what clues do I have that that's the level I'm at? Well, it tells us that the epimesium is surrounding it. I see evidence of that deep fascia continuing down to the tendon here. And it does label that this portion pulled out is a fascicle, then the muscle cell being pulled out. So this looks like a bunch of straws within straws, right? And it can be difficult sometimes to figure out which layer you are looking at. So I think looking at these three different images plus the Blossom images and looking at the animations, just trying to get a sense of these layers of layers of layers. But I hope you're walking away appreciating that a muscle is a bundle of bundles of bundles, right? Of structures, and that there are lots of blood vessels and nerves that travel down through these connective tissue layers down to the individual muscle cells. And remember that the individual muscle cells are able to contract. What you will learn is that when muscles contract, the entire muscle, when the muscle cells contract, the entire muscle also contracts and changes shape. So again, other views of this, everything's labeled up for you. I like this particular image because it shows us that this is a fascicle. We see the perimysium stripped away, but we also see a beautiful rendition of arteries, veins, and nerves deep within the tissue. And then again down here, this is a muscle cell. We see the outer layer of the endomysium. We see the pulled out myofibrils. We see the satellite cell. And this image labels, once again, the sarcolemma, which is the outer cell membrane of a muscle cell. So in addition to, let's make sure you get this, there is an endomysium, that is the outer connective tissue layer. But deep to that, there is a cell membrane of the muscle cell, and that is specialized name sarcolemma. Inside the cell, rather than calling it a cytoplasm, and you know that term, the cytoplasm is simply the cytosol with all of the organelles, because the muscle cell is just so different and is full of these myofibrils. The cytoplasm is instead called sarcoplasm. Sarcoplasm, so sarcoplasm rather than cytoplasm. Heading back to the Blosson, and there's another term here that I've introduced in the lab, and that is aponeurosis. And aponeurosis is a broad, tendon-like sheet of tissue that connects muscle to muscle. And aponeuroses are made of the same exact stuff as tendons and ligaments, and you know that that is dense, regular connective tissue. In the lab, you have seen the linea alba. You know that there are 4 layers of abdominal muscles. There are no bony connections over your gut. So those 4 layers of muscles, the rectus abdominis, the obliques, and the transverse abdominis, those muscles must all be attached to each other, and they are through various aponeuroses. In the lab, you learn the linea alba. You know that your left and right rectus abdominis muscles are connected mid-sagittally by a white line. That white line is actually an aponeurosis. So there are a couple of aponeuroses you're learning in lab. The other one you're being asked to know is the epicranial aponeurosis. That aponeurosis, that thin sheet of tendinous-like material that goes over the cranium, epicranium, and connects your frontalis to your occipitalis muscles. So let's dig into skeletal muscle fibers. We're looking down at the cellular level now. Again, keep in mind your muscle fibers are very, or can be, they don't have to be, but they are very long cells. And for example, we're talking about your sartorius. Sartorius is a significantly long muscle. In a taller person like me, it's well over a foot in length. And that means that your muscle cells are as long as the muscle is. So we're talking about hundreds and hundreds and hundreds and hundreds of cells linked end to end, maintaining their nucleus and allowing large amounts of proteins and enzymes being produced along the length of that muscle for muscle contraction. I've already introduced the term sarco, meaning flesh. And we've seen that sarco term used in describing the sarcolemma, the cell membrane of a muscle cell. We've seen sarcoplasm, the cytoplasm of a muscle cell, and one more specialized organelle that you've not heard about before, and that is sarcoplasmic reticulum. Now, the sarcoplasmic reticulum sounds a lot like the endoplasmic reticulum. This is a specialized, smooth endoplasmic reticulum found in muscle. And I've told you before that muscle requires buckets, that's my word, right? Buckets of calcium, lots and lots of calcium to do their job. Well, not only does bone release calcium, but muscles must also store calcium internally so they can do their work. And so the sarcoplasmic reticulum is a network of membranes in the muscle that store and release calcium as needed for muscles to do their contractile work. And then lastly, another term with sarco in it, there is the sarcomere. And the sarcomere we'll see is the functional unit of skeletal muscle cells. And that will be something we'll be talking about here quite a bit. As we look down at this animation, definitely watch that. Let's get down to this picture. This is a zoomed in illustration of a muscle cell. And let's just take a look at what we're seeing. The cell itself has its own cell membrane, but notice this cell membrane has some holes in it. It's a very different looking cell membrane than what we're accustomed. We won't be dealing with the specifics as to why, but this sarcolemma, the outside layer, is so unique that we've given it a different name. Those openings are critical to how the nervous system interacts with muscle and causes muscle to contract. That will not be a featured conversation this semester. That'll be a physiology concept. But that's the sarcolemma. Inside the cell, we see it's stacked full of these myofibrils. Not only is the muscle cell long, but these myofibrils run the entire length of the muscle itself. Also, shoved inside the sarcoplasm, right, the cytoplasm, we see tons and tons and tons of mitochondria. Well, we're not surprised that there are lots of mitochondria in a muscle cell because we need lots of ATP. We need a lot of energy to do the work of contraction. The other thing I note that's very unusual looking here is that there's this web, this network of membranes all along the myofibrils. This purple web is the sarcoplasmic reticulum, right? So the sarcoplasmic reticulum that I just mentioned is that network of calcium storing membranes. And here's where the calcium is being stored. And you see it, it's just everywhere. It just a bunch of webbing everywhere. And we're not going to be breaking this down into greater detail, but you can see here it's labeled the sarcoplasmic reticulum. And we are not worrying about T-tubules, terminal cisterna, or triads this semester. So that fact and any of those questions related to T-tubules can be skipped for now in this semester. So as has become our custom, we've now looked very specifically at the cells that make up muscle tissues, these skeletal muscle cells, and we have to go even deeper to understand how muscles do their work. So now let me introduce to you the idea of a sarcomere. A sarcomere is really what gives the striated appearance of skeletal muscle cells. And a sarcomere means part, a unit. So if I were to say to you, what is a polymer? It is a molecule made up of many parts, many pieces, many subunits. If I said, what is a dimer? It is a molecule made up of two parts. If I said a monomer, it's going to be made up of one part. So a sarcomere, is simply a part of the muscle. This is the, quote, functional unit. If we were back in compact bone, I would say that the osteon is the functional unit. We spend a lot of time looking at the osteon and the structures of the osteon. That is the functional unit of compact muscle cells. And now we see the functional unit of skeletal muscle cells. We're not going to be discussing troponin and tropomyosin this semester. These are other proteins found in the sarcomere, but we won't be focusing on those. Again, those will be proteins that you will learn very much about in physiology. So let's take a look at the sarcomere. There's some really, really good animations here to help you with this. Definitely watch this, but before you watch that, let me try to give you a breakdown in this image first. and that is along the myofibril, right? Each of those myofibrils is composed of end to end sarcomeres. So when you look at a myofibril, let's just scroll back up, right? Here is, we see that inside the muscle cell, there are these myofibrils, and we see the alternating light and dark pattern. What is making those alternating light and dark patterns are sarcomeres end to end down along the entire muscle cell and down along the entire myofibril. And what is making these up are proteins called myofilaments. So we have myofibrils with lots of myofilaments. Myofilaments, when I hear the word filament, I think of the tungsten filament in old incandescent light bulbs, right? That little tiny thin wire that you find inside light bulbs, old fashioned light bulbs, a very thin filament. And filaments come in two flavors, actin and myosin. Actin is the major protein and it is called the thin filament. Myosin is the protein found in the thick filament. So there are thick and thin filaments that are running along the length of the sarcomeres and the myofibrils. Let's take a look at this cartoon representation of this. So this is just one sarcomere. A sarcomere goes, its edges are at what's called the Z line. A Z line is a protein, but they call it the Z line, and it's shown here in this Shrek looking green, right? So the Z line from Z to Z is the sarcomere. We see in the center of the sarcomere this thick filament. This is the myosin. It's shown here in this orangey red. It's a big filament, thick myosin. And then we also see these thin purple filaments. These are the actins. So you see the thin filaments and the thick filaments. And you see that there are areas where they overlap. Now we're beginning to get a glimpse of why we see striations. We see alternating light and dark regions under the microscope when looking at both skeletal muscle and cardiac muscle. So zooming in, right, the red is the thick filament, the myosin, and the purple is the thin filament, which is primarily made up of this blue protein here called actin. There are some other terms used to describe the sarcomere, where the myosin is. So this entire area where the myosin is also referred to as the A band. I remember this because A is a wide letter. It's a thick letter. And so the A band is where the thick filament is, the myosin. Where there is just the actins, where there's just actins, that's the I band. Well, I is a thin, skinny letter. So the I band is where the thin filament is. Again, just use those little analogies, those little connections to remember this. The A band, thick filament, the I band, the thin filament. Right down the middle, there's an M line. M line, middle, M, easy. Okay, easy to remember. We won't be going into the specifics of the myosin molecule or the specifics of the actin molecule. Just see that they attach to each other and they pull on each other. So when a muscle contracts, what's happening is that the actins and myosins are grabbing onto each other like a bunch of people playing tug of war side by side, and they're pulling each other closer to each other. And you're going to have, therefore, this whole sarcomere is going to get shorter, more compact. And what we don't see in this picture is that there's another sarcomere to the left and another one to the right, and beyond, there are hundreds, thousands of these sarcomeres all in a row. They're all shortening at the same time. And that is the magic that allows the muscle to contract. You'll hear about the sliding filament theory as the way in which muscles contract. Sliding filaments, what they're talking about is that sliding myofilaments, these actins and myosins sliding over each other to contract the muscle. So definitely watch the animation. It'll really assist you in appreciating how this works. And then we're going to stop right here. I'm going to mention this term, neuromuscular junction. And that is simply, I told you that the nervous system interacts with muscle cells, telling them to get excited and to contract. Well, the where the neuron, where the nerve cell connects to the muscle cell, that is referred to as the neuromuscular junction. Just know that term, the neuromuscular junction, where the nerve and the muscle join, and you will learn lots and lots and lots about how the neuromuscular junction works in physiology. That is, how does the nerve excite the muscle and what is involved in that interaction. We will be looking at the nervous system, and I'll mention that NMJ again, but again, we won't be getting into the details of how it works, but I will describe that phenomenon a few more times when we get to the nervous system and how the brain and spinal cord interact with your muscles. And then as we scroll through this little section on neuromuscular junction, the next section, excitation, contraction, coupling, notice I've made it blue. The rest of the chapter, the rest of the section, you can skip. We're not doing any of this material. However, at the end of this section, 10.2, you are able to answer a couple of questions. That is, you can answer questions 3, 4, and 5. You should also be able to label the majority of the muscle cell. And you should also be able to tell me about the 3 levels of connective tissues in the muscle. So don't completely ignore the terms here, and also the vocabulary exercise here will be helpful. The rest of the chapter we are not going to be dealing with. So 10.3, 10.3, 10.4, 10.5, 10.6, we're skipping over those. In a moment, I will go to 10.7, and I will go to a moment at 10.8. Going back to the PowerPoint slides for a moment, I've got 3 or 4 pictures here that give you a static appreciation of how the sarcomere works. Again, we see the sarcomere here drawn out. We see the overlapping thick and thin filaments. We see the A band labeled, the I band labeled. And under the microscope, or under an electron microscope in this case, you can clearly see the overlapping light and dark regions. So just follow it straight up, and you realize that the darkest bands are where the actins and myosins are overlapping. And the lighter colored bands are where there isn't the overlapping, just the thin filaments here, giving us a lighter color. And then you can see the dark band also of the Z disc at the ends of the sarcomere. Well, what happens when this now begins to contract? So in relaxed muscle, under the microscope, you would see this. Again, the actins and myosins are slightly overlapped. But as that muscle begins to contract, you see that the filaments are sliding over each other. As the filaments slide over each other, there's going to be even more dark areas because of the increased overlapping of the actins and myosins. If you were going to shine a flashlight right through this area, there'd be less light able to get through it, right, because of the overlapping. So you have a darker region. And notice that as the filaments slide over each other, the Z to Z lines actually get closer to each other. So the entire sarcomere is shortening. Then let's go one more step and see that when the muscle is fully contracted, look at the picture up here, the biceps is now completely contracted. You're lifting something as heavy and hard as you can. All of the actins and myosins have overlapped to their maximum capacity. The sarcomere is much shorter. The band would be mostly dark. And imagine if all the sarcomeres along the muscle are now as short as they possibly can, right? You have shortened that muscle as much as possible. You've exerted all the force that is possible from that muscle. So it's kind of fun to just kind of look through these slides back and forth and just kind of imagine if you could see under the microscope how the sarcomere changes during contraction. You can kind of mimic that by just going back and forth through these slides, or better yet, watch that animation in Blossom. Okay, so we're jumping down to 10.7, covering cardiac muscle tissue. This is going to be largely a review of things you already know about cardiac muscle tissue. So let's take a look here. So you know that cardiac muscle, so we're flipping from skeletal muscle and we're now comparing and contrasting cardiac muscle. And you know that cardiac muscle is only found in the heart. You know that it is constantly working, right? It's a continuous muscle that must continue to work our entire lifetime. You know that it is composed of striated cells that are, and because they're striated, you now understand that they have actin myosin that is arranged in sarcomeres just like skeletal muscle. But one of the differences is that cardiac muscle cells are shorter and they usually only have one nucleus, whereas remember, skeletal muscle is multinucleated, and they are branched, right? They are branched. Like skeletal muscle, cardiac muscle also has lots and lots of mitochondria because we are constantly needing lots and lots of ATP for these cells to do their job. So I really want you to be able to compare and contrast cardiac and skeletal muscle cells very, very clearly. And finally, you know that in cardiac muscle cells, there are these things called intercalated discs, and you'll understand a little bit better today why those little discs are important in the work that the heart does. So let's talk about those intercalated discs. Let's take a look quickly at a cartoon of cardiac muscle fibers. So in this diagram, we are seeing, right, this squiggly line. This squiggly line represents the intercalated disc. Again, we see lots of those mitochondria embedded in the muscle cells, and we see that there's not multiple, but just usually only one nucleus per cell. We also see the branching, right? The branching of the cells. So under the microscope, we see these strong lines. These strong lines are the intercalated discs. Well, what in the world are they? If we look now, this squiggly white line represents an intercalated disc between adjacent adjacent cardiac muscle cells. Well, what is an intercalated disc? It's actually two things that you already know about. It is a combination of desmosomes and gap junctions. We learned about both of these back in the epithelial chapter. We learned that desmosomes are reinforcement buttons or rivets, that these connectors are found between cells that are under a lot of stress. Well, imagine your heart is beating 60, 70, 80, 130 times a minute when you're exercising. And so those muscle cells are under a lot of stress and they're being pulled apart as they contract. So we need those reinforcements. And gap junctions are a little channel that not only connect the cells together, but also allow for the sharing of information between the cells. And those gap junctions in your heart are not only allowing nutrients to move through, but also the electrical signals to travel through the heart super, super fast. You want all of the cells in your heart contracting as a unit, as a single unit, so that the heart can pump very, very, very strongly together. And that idea is right here, mentioned here, in that these the gap junctions are allowing a way for the electrical signals to quickly transmit from one muscle cell to another muscle cell in the heart, so that the muscle of the heart contracts as a unit. We want all of the cells contracting at the same time, and that's called a syncytium. Look at that word. And what you're seeing is basically three words thrown together that you know. Syn, right? Coming together, sync, synchronized, and cyto cells. So syncytium is really just a really cool word of really three terms thrown together, smushed together. Syn for together, sync, right? Think synchronization, working together, cyto cells. So the synchronized contraction of the cells, right, is really what's happening in cardiac muscle tissue. And that's it, guys. That's really it. One more thing about cardiac muscle cells is cardiac muscle cells have within them, or there are within the heart, specialized cells called pacemaker cells. You've heard of people having a pacemaker put into their body. A pacemaker is an external device which is zapping your heart, telling it to contract. But your heart has this naturally. So in your heart, you already have these pacemaker cells. Those pacemaker cells actually set the rhythm of your heartbeat. And you may not realize this, but your brain does not send signals to your heart telling it to beat every single time. Your heart does that itself. So we say that the heart is autorhythmic. Your heart has the ability of firing itself, of maintaining its own rhythm. Yes, your brain can influence your heart rate. It can cause your heart rate to go up or heart rate to go down. But your heart itself is autorhythmic, setting your base heart rate. And then thirdly and lastly, we know, or you know, that calcium is incredibly important in muscle contraction, and that is no different here in cardiac muscle. We need lots of calcium to do the work of muscle contraction. And that brings us to the end of cardiac muscle. Very quick review of that tissue, a few questions for you to answer, and then we're going to jump down to smooth muscle 10.8. Again, your job is to be comparing and contrasting these tissues. And remember, smooth muscle is so called because we do not see striations. Now, smooth muscle still has actin and myosin, but it isn't arranged in sarcomeres, and therefore you don't see the striation patterns. Where do you find smooth muscle? You find it in the walls of your hollow organs. Think your bladder, uterus, stomach, intestines, walls of passageways. Think your arteries and veins also have smooth muscle, as does your respiratory tract, okay? Along the fallopian tubes, along the vas deferens of the male. Again, lots and lots of smooth muscle pushing things along. You also have smooth muscle in your eyes. You know that your pupil changes shape. You have no control over that. So that's a good clue that your pupil, the muscle that opens and closes to let more light in your eye, that muscle is smooth muscle. And finally, you know that you get goosebumps, and that from the skin you learned about the erector pili muscles. Those muscles are also smooth muscle. We have smooth muscle all over the place, right? All over the place. But remember, it is involuntary. the picture here is showing us that the cells are individual. They are cigar-shaped cells, or what we call fusiform in shape, and they are not striated, but instead, when these cells are contracting, watch the videos, but when these cells contract, here is a relaxed muscle, a smooth muscle cell, and here is a contracted muscle cell. We still see that there is a shortening, a changing of the shape, but again, there are other things that cause that to happen. We're not going into dense bodies. We're not going into the intermediate filaments, but these are structures that help with the contraction of the smooth muscle cells. All this blue stuff we're not worrying about. Okay. How are smooth muscle cells the same? They still rely on calcium. They still shorten, but it's different. They have a single nucleus and they are fusiform in shape, which is a fancy way of saying that they are cigar shaped. Let me go ahead and write that word because I don't see it here. And the shape we would say that these cells is fusiform. Fusiform. And that brings us to the end of what I want you to know for smooth muscles. So the rest of it's all blued out as a reminder that we're not focusing on that. Ooh, one more last thing, and that is this. And this is a similarity, is that skeletal muscle and cardiac muscle and smooth muscle can all undergo hypertrophy. Hypertrophy is a term that means increase in size. If you work out in the gym, your skeletal muscles are increasing in their size. They're undergoing hypertrophy. But another thing that smooth muscles can do that is unique is that they can produce more cells. And this is the term hyperplasia. Hyperplasia. And I only want you to think about this in one situation, and that is during pregnancy. The uterus is lined by a layer of muscle called the myometrium, right? Myomuscle, metri means uterus. So the myometrium is the muscle layer of the uterus. And during pregnancy, the number of cells that make up the uterus increase tremendously. Not only do they get bigger in size, there's also a greater number of cells, and a greater number of cells producing more cells is called hyperplasia. So hypertrophy is an increase in size. Hyperplasia is an increase in the actual number of cells. When you work out in the gym, you do not get more skeletal muscle cells, but each of those muscle cells gets bigger, right? So in the gym, you undergo hypertrophy of your muscle cells, but you don't actually get more muscle cells. It's just that each of the cells you already have get larger. And that brings us to the end of the smooth muscle section. And finally, we're going to jump over very briefly to 11.1, the very beginning of chapter 11. And these are all facts that you've already learned in lab. I'm just going to mention them again here in 11.1. And that is, you are learning about origin insertion. How is it that muscles are attached to your skeleton, and this idea of origin and insertion? So let's just remind ourselves, in the biceps brachii, and the biceps brachii, you break it down, right? Bi means two or double, and seps means head. So we're talking about a double-headed muscle in the brachial region. Sure enough, the biceps brachii has two heads, or two portions of it. We're not asking you in lab to know the difference in those two heads. There's a long head and a short head. We're not learning about that, but just know that it is a double-parted muscle. And you are being asked to know in the lab that it attaches up to two places in the glenohumeral joint in the shoulder, one to the coracoid process and one to the supraglenoid tubercle. of the scapula. And you're also being told to know that it attaches down to the radial tuberosity. But you also need to learn which of those two locations is the origin and the insertion. Recall that the origin is the place where, when the muscle contracts, it doesn't move very much, and the insertion is the place that moves the most. So as you contract your biceps brachii, the insertion, the radial tuberosity, is moving up toward, it's moving, whereas your shoulder is not moving. So that means that the coracoid process and the supraglenoid tubercle, these are your origins of the biceps brachii. The radial tuberosity is your insertion. So make sure you understand the origin and the insertion as a concept for the lecture exam, and then the lab exam, you're going to be asked about quite a few of these specific origins and insertions. Another concept that was mentioned in the lab is that there's prime movers. Prime movers are the main muscle that does something, right? So in this action, the prime mover is the biceps brachii as we flex the elbow and lift up this cup. But there's other muscles that also assist in this movement. And here the book shows us the brachioradialis is a synergist. It helps in the process of flexing. So we see brachioradialis must be a muscle that goes from the brachium, right, up in the brachium down to the radius, right? Don't make it too hard. And the brachialis is also another synergist in this movement. So the brachialis is a muscle that is deep to the biceps brachii. You can see here the biceps brachii has been cut away. We see the two tendons, but the brachialis is deep to the biceps brachii and is assisting also as a synergist in the movements. So make sure you understand origin, insertion. Also, make sure you are reviewing your movements like flexion and extension. And then one more thing is that muscles have their power when they shorten. We talked about how muscles contract, one of the characteristics of muscle tissues. So when a muscle contracts, that is where the power comes in, but there's always a opposite motion, and the opposite motion is always done by an antagonist. So an antagonist is the muscle that does the opposing work. You might see it described as an antagonist. You might see it described as an opposing muscle. And in lab, you have learned about a couple of antagonistic muscles or opposing muscles. You've also learned about an opposing muscle groups. So you know, for example, that the flexion of the knee is the job of the hamstrings, and the extension of the knee is the work of the quadriceps. So your quadriceps femoris muscle group and your hamstring group are opposites or antagonistic to each other. You also learned in lab, for example, about the extensor muscles of the wrist extensors and the wrist flexors. Those are also opposing muscle groups. In lab, you also learned about the sternocleidomastoid and the splenius capitis. The sternocleidomastoid lets you flex your neck. The splenius capitis allows you to hyperextend your neck. Again, those are opposing muscles. So you've learned about some of these opposing groups and opposing muscles. Make sure that statement and that understanding is clear for you. And here's a couple of examples of antagonistic muscles. And that pretty much brings us to the end of this little section. Oh, one more thing. I do want to remind you that muscles come in multiple shapes. Again, this was introduced in the lab presentation for back in lab 4 and 5 presentations. We know that muscle fibers appear in groups or fascicles, and those fascicles can be in different shapes. Let's start at the top here. We have circular muscle groups. You have seen in lab two circular muscle groups, the orbicularis oris and the orbicularis oculi, around the lips and around the eye. Muscles can be long and parallel, so we say they're parallel. You can see muscle groups that are convergent. They are sort of triangular shaped, and you can see the fibers are moving, they're converging onto a common place. Your pectoralis major is a good example of a convergent muscle. Then we have pennate. Pennate means feather shaped, and you can see bipennate. Rectus femoris is a good example of a bipennate muscle. You see the two sides of the feather looking structures. We have unipennate. I'm not going to ask you specifics on these, but pennate just means feather shaped looking muscles. And then multipennate as well. The deltoid is an example of that. Here's that word, fusiform, right? Fusiform is the idea of a smooth muscle cell, or even a muscle like the biceps brachii, where there's two narrow tendons at the end. We would say that that muscle is fusiform in shape. And this just goes through the different shapes and describes them to you more completely. I put a blue line right here. The rest of this chapter we are not covering, so don't worry about the lever systems. And down here in the questions, you can certainly answer a few of these questions about the synergist. You can answer questions about contraction and extension, but don't worry about muscle questions related to don't worry about any muscle questions about the levers. So that brings us to the end of all things in the Blosson as part of the muscular system presentation. I'm now going to go back to the last few of those PowerPoint slides. Okay, so jumping back into the accompanying PowerPoint slides for the muscular system. The very first slide, just a reminder, I went through this before as well, but on the exam, there will be connection questions. Connection questions are meant for you to appreciate how one body system is connected to or interacting with other body systems. This particular slide goes through examples of how the skeletal system is interconnected to other body systems. I want you to also be thinking about how the muscular system speaks to other systems as well. On the exam, there will be 6 questions, 2 for the integumentary, 2 for the skeletal, and 2 for the muscular system, where you will need to list a very specific interaction. Don't tell me that the skin covers the muscles. You knew that before you came into this course. Don't tell me, for example, that muscles interact with the bones. That's too simplistic. Things like muscle cells require calcium, which is stored in the bones in the calcium phosphate inorganic matrix of bones. Something like that. Or that osteoclasts, when they resorb bone, are releasing calcium, some of which is necessary for muscles to contract. There is a well-written statement that connects the two systems together. So, as you're going through the lectures, think about 2 other systems that I talk about the integument, 2 other systems that work with the skeletal, 2 other systems that work with the muscular system as well. So we've already talked through these slides. Back about cardiac muscle. You know that these are found only in the heart. You know that they are shorter. You know that they have usually only 1, sometimes 2 nuclei. You know that these cells are branched and they have intercalated discs. And I introduced the idea that these cells are also autorhythmic in that they generate their own pulse or their own contraction cycle without the need of the brain sending those signals. You've seen them under the microscope. Here's another cartoon representation. Again, we see the Y branching, we see the striping, we see the intercalated discs, we see usually a single centered nucleus in these cells. Those are the characteristics of the cardiac muscle cells and tissue. Also, quick reminder about smooth muscle. These are short muscle cells. They have a central single nucleus. They are fusiform in shape. Yes, they still have thin and thick filaments, but they're not aligned in sarcomere arrangements. Therefore, we do not see striations. Smooth muscle contracts more slowly. It is resistant to fatigue. This is the kind of muscle that just keeps on going. This is the tortoise and the hare. The tortoise just keeps on going. That's a good analogy for smooth muscle. Because it isn't as fast, it doesn't require quite as much ATP as skeletal muscle or cardiac muscle. So that also means that smooth muscle would have fewer mitochondria within the cells. The contraction is slower. The relaxation is slower. And don't forget that smooth muscle, like cardiac muscle, is involuntary. Again, comparing and contrasting the muscle types. Again, we're not worried about the dense body sort of structures. They still have thick and thin filaments in them, but not arranged in the same way. And the cell does absolutely change shape. We go from this long, skinny, fusiform shaped cell to a scrunched up, contracted cell. You already know the functions of skeletal muscle, and I've already gone over the 4 characteristics of muscle tissues. Muscle tissues are excitable, contractile, elastic, and extensible. And we've already talked about agonists and antagonists, synergists, prime movers, and I also introduced in 11.1 the idea of the origin and insertion. We've already been through these slides earlier in this presentation. And origin, remember, is the place that doesn't move. Zero movement. Oh, origin, zero, null, less movement. Whereas the insertion moves with the contraction of the muscle. We've already gone through the sarcomere slides. And that brings us to the very last thing. What happens when death occurs? You've heard of rigor mortis. And oh, as a sideline, no cats were harmed in the making of this video. I don't know if this cat's dead or if it's just stretching. My cat looks like this 3 times a day as it's waking up. However, a rigor mortis is simply what happens to the muscle tissues following death. And what happens is that there's no longer oxygen, right, going to the body. And it takes ATP, it takes energy for your muscles to contract and to release from each other the actins and the myosins. So shortly after death, the muscles become stiff, and it stays that way to about 36 hours after death. Now, at 36 hours, what's happening is that the cells actually are undergoing an autolysis reaction, right? They're undergoing an autolysis. They're breaking down. So the lysosomes inside your cells are releasing their products, and all of those digestive enzymes are beginning to break down everything in the cells as the cells begin to decompose. And in that decomposition reaction, the muscle cells are beginning to relax and release as part of that autolysis reaction. What do we have to look forward to as we get older? Our skeletal muscle mass will become less, right? Our muscles will undergo atrophy. We will lose power and size of our muscles. Now, of course, we can slow down this process by continuing to exercise, but over time, a lot of our muscle mass will be replaced by adipose, right? We become a little bit flabbier with age, and also with some fibrous connective tissue. And what that's going to do is make our muscles a little bit less elastic, right? They're not going to be as supple in movement. They're going to be a little bit more fibrous, a little more dense and tough. Our strength will decrease. And don't forget, that includes our heart. Our heart is a muscle, so cardiovascular, also our cardiovascular cardiac muscle function will decrease. That means that we'll have a little bit less blood flow going to our muscles. Our ability to exercise will decrease. Our tolerance for exercise decrease. And when we do exercise, we will be more prone to fatigue and it will take us longer to recover. This is obvious. A 12-year-old can go out and play soccer for a long, long time. They don't seem to ever get tired and they don't need much time to recover. However, if I go out and I play soccer, I'm not going to do it for very long. I'm going to fatigue much more rapidly, and it's going to take me months to recover from that soccer game. So we know this happens normally. And as I said earlier, your muscles start losing elasticity. So we're a little bit stiffer when we wake up in the morning, and we certainly are not as limber and don't have quite the flexibility. That's a combination of muscles and joint changes. So that brings us to the end of all things muscle that I want you to know for exam number 2.